Hey everybody, welcome back. Today we're going to be talking about nucleotides, nucleic acids, and genetic information. So let's go ahead and get started. So here are our learning objectives for this chapter. So make sure, read over these learning objectives, keep them in the back of the mind as we go through this lecture and as you read the material. And afterwards, come back to them and make sure that you can do all of these learning objectives that I had listed for you. All right, let's go ahead and get started. All right, so nucleotides are composed of nucleotides. So that's supposed to say nucleic acids are composed of nucleotides. All right. So nucleic acids, it's very obvious. We know that nucleic acids such as DNA carry genetic material, store genetic material, and then we use RNA to transcribe and translate DNA in order to make proteins. What is less commonly known is that nucleotides also facilitate and participate in oxidation reduction reactions, energy transfer reactions, intracellular signaling. They also play a role in biosynthetic reactions, as well as the storage and decoding of genetic material. Nucleic acids have also been known to play some structural roles, as well as in catalysis, specifically with RNA molecules. And we'll come back to that in just a minute. So if we look at the different layers, I guess, so to speak, of what makes up a nucleotide, first, if we have just the purine or the pyrimidine, we have a nitrogenous base. Once we add the sugar, we get a nucleoside, and then once we add our phosphate group, we have a nucleotide. Now, we can have multiple iterations of nucleotides because we can have a monophosphate, a diphosphate, or a triphosphate nucleotide, depending on how many phosphate groups we have. So just keep that in mind. When the phosphate group is attached, we have a nucleotide. If we have just the sugar, it's a nucleoside, and if it's just the base, it's a nitrogenous base. Now remember, we have 5 basic nitrogenous bases. There are a couple others that are less common, but these are your most common nitrogenous bases. We have the 2 purines, which are adenine and guanine, and the 3 pyrimidines, thymine, cytosine, and uracil. The types of bonds that are found in nucleic acids. So when we look at our nucleotides, between the nitrogenous base and the ribose, or the deoxyribose, if we're talking about DNA, we have a glycosidic bond. And between the sugar and the phosphate, we have an ester bond. And we see that the ester bond occurs at the 5' carbon on the sugar, and the glycosidic bond occurs on the 1' of the sugar with the 9 nitrogen on our, for this instance, purine. If we look at a polymer of nucleotides, say in a strand of RNA, we also have phosphodiester bonds linking the 2 nitrogenous or nucleotides together. Notice at the 5' end, we have a free phosphate group, and at the 3' end, we have the free hydroxyl group. Your phosphodiester bond is going to occur between the 3' sugar for the phosphate and then your 5' carbon on the sugar on the other side of the phosphate group. Another bond that we find in nucleic acids that we've talked about on a number of occasions is hydrogen bonding between our base pairs. So when we have a GC base pair, we get 3 hydrogen bonds, and then on the thymine adenine base pair, we have 2 hydrogen bonds. So these are all the different types of bonds that we can see in nucleic acids. They all have different energies associated with them, and they all require usually different enzymes to help facilitate the bond formation. And, oh, there's my pen. I lost my pen. Okay. And we'll talk more about that in later chapters when we talk about DNA metabolism. Okay? So let's talk about the differences between purines and pyrimidines. So here we have our purine. And our numbering scheme is, as you can see here. So we have the nitrogen 1, and then we count clockwise, as seen here. So we get 1, 2, 3, 4, 5, and 6 here at this carbon. And then we continue counting on our 5-member ring with 7, nitrogen 7, carbon 8, and nitrogen 9. Now remember, when we add our sugar gets bound here at this nitrogen on a purine. The reason I want you to remember the numbering scheme for these molecules is because if you know the proper IUPAC naming of the bases, you can figure out their structure. So for instance, the 6-aminopurine, which is also called adenine, we can see that on carbon 6, right here, we have an amino group. For guanine, its proper name is 2-amino-6-hydroxy. So as we can see on our carbon 2, we have our amino group, and on carbon 6, we have that, it's either going to be formed, depending on the pH, a double bonded oxygen, and we can also have a hydroxyl group, which is why it's called 6-hydroxy. But at the pH that we are in the body or in the cell, normally that is deprotonated. Plus we have, what's the word I'm looking for? Resonance here with our double bonds, our conjugation that's going on. Okay? So that's why it's important for y'all to memorize both the IUPAC name as well as how the rings in the purines and pyrimidines are labeled. Here, if we look at our pyrimidine, we start with N1 down here, and then go clockwise, 2, 3, 4, 5, 6. So thymine being 2,4-dihydroxy-5-methyl. We can see that on carbons 2 and 4, we have our oxygens, and then on carbon 5, we have a methyl group. Cytosine being 2-hydroxy-4-aminopyrimidine, we have our 2-hydroxy group, and then our 4-amino group. Uracil is the same as thymine, except we do not have that methyl group here on the C5, but we do still have the 2,4-dihydroxy groups. Okay? So take some time, practice that out, drawing it out, and learning the numbering scheme and memorizing the names. This way you don't have to memorize 1, 2, 3, 4, 5 different bases. You just have to remember the purine, the pyrimidine, and then the naming. And once you can do that, you can draw any of the bases. So our differences between ribose and deoxyribose. First off, both of them are 5 sugars in a 5-member ring that is heterogeneous in that we have an oxygen in the ring instead of a carbon. So we have C1 prime, 2 prime, 3 prime, 4 prime, and 5 prime is up here. The reason we give it the prime terminology is to differentiate it from the nitrogen bases that just use 1, 2, 3, 4, and 5, so on and so forth. So if we're talking about a 3 prime sugar, then we mean the sugar on the, if we're talking about a 3 prime carbon, then we're talking about a carbon on the sugar, not a carbon, say, on the pyrimidine or purine, which happens to both be nitrogens, but you get the point. So primes for the sugars, non-primes for bases. Obviously, the difference between deoxyribose and ribose is a hydroxyl group on the 2' sugar for ribose, and that gets replaced with just a hydrogen on the 2' sugar for deoxyribose. So we've lost a hydroxyl group. When you're thinking about this, and this does play a role, by replacing that hydroxyl group with a hydrogen, we've messed around with the polarity a little bit, because now we're one less polar functional group, and the reactivity of this molecule has changed. And we can talk about that a little more in class, about the implications of that as far as DNA degradation versus RNA degradation. Because of this hydroxyl group, ribose is more likely to degrade than deoxyribose. So deoxyribose is more stable because we've lost that hydroxyl group, which makes it less reactive compared to ribose. Okay? So, when we're talking about nucleotides, we have to talk about ATP, or adenosine, right? Because we have an adenine nitrogen base. Because we have 3 phosphate groups. Okay, so adenosine triphosphate. And the reason adenosine triphosphate is so important is because it is the most commonly used nucleotide in energy transfer reactions. So we see it all the time in glycolysis, the citric acid cycle, and oxidative phosphorylation. So this is actually the molecule that gets phosphorylated. We add a phosphate group, say from the ADP to make ATP, or AMP to make ATP, in oxidative phosphorylation. And the reason it's such a good molecule to transfer energy is because these bonds here, the POP bond that we have, the phosphate diester bond that we have here is or phosphate anhydride, I'm sorry. Let me make sure that's the correct terminology that I'm trying to. Yeah, phosphate anhydride. Okay, these are very high energy bonds. So if you see here, if we hydrolyze ATP into ADP plus inorganic phosphate, we get a delta G of negative 30.5 kilojoules per mole. So as we hydrolyze this bond, and I've written over this a lot, we release 30.5 kilojoules per mole of energy, and that's a lot of energy. Now, furthermore, if we hydrolyze ATP using water to form AMP and pyrophosphate, which is the PPI, we get negative 32.2 kilojoules per mole release. So it's very exergonic. We're releasing a lot of energies. Furthermore, the pyrophosphate can be hydrolyzed to release an additional 33.5 kilojoules per mole. So if we add these two together, so the hydrolysis of ATP to form AMP and 2 inorganic phosphates, we end up with a total of approximately 60 kilojoules per mole of energy released. So this is the molecule most commonly used, say, to release energy. So we have food molecules plus oxygen to give us CO2 and water, which drives, so this is oxidative phosphorylation or oxidative respiration, which will then form ATP by bringing ADP and phosphorylating it to make ATP. And then furthermore, we can then use the ATP generated through oxidative phosphorylation to drive, say, biosynthesis. Say we're making a protein and we require ATP in order to synthesize that protein. So from reactants to products, we can drive that reaction forward through the release of energy of ATP. And we'll talk more about that in metabolism and glycolysis, and you'll really see where ATP is used. But I just wanted to point it out here in this chapter because since we are talking about nucleotides and nucleic acids, ATP is a nucleotide. So why is ATP hydrolysis so exergonic? Again, if we do ATP to yield AMP plus 2PI, we end up with about negative 60 kilojoules per mole total. Okay, so it's actually two reactions occurring, but the sum of that energy is about negative 60 kilojoules per mole. So one of the reasons is at physiological pH, ATP itself carries 3 to 4 negative charges that repel each other. So hydrolysis of ATP to either AMP, which is 1-phosphate, or ADP, either one, both of those reduces those electrostatic repulsions because you reduce the number of negative charges on one molecule, which makes it more soluble in water and a happier molecule. Also, the resonance hybridization of the products of ATP are more stable than ATP. As you can see here, we're able, if we just have one phosphate group, you can see we're able to have more resonance structures than if we had the 3 phosphate groups attached. There's actually less resonance capabilities. And then again, you can see here negative charge, negative charge, negative charge. The products of ATP hydrolysis are more soluble in an aqueous environment, again, because we are distributing that negative charge across multiple molecules as opposed to just one molecule, and they're smaller. Also, because we're breaking one molecule into 2 or 3 molecules, we have a positive delta S, increasing the entropy of the system. All right, so now let's get back to DNA. So the double helix. As you can see here, the double helix consists of 2 strands of DNA that base pair in a complementary fashion. And this was first discovered by Edwin Chargaff in the 1940s, where he found using UV vis and paper chromatography, which I'm sure all of y'all have done paper chromatography before. He was able to show that the concentration of adenines is equal to the concentration of thymines, and that the concentration of guanines were equal to the concentration of cytosines. This was one of the first indications that DNA may actually carry the genetic material. Before, people thought that these bases were just in random mixes, that they didn't really make it, mean anything. But now that we know that there is some complementary, they're starting to wonder maybe there's more to DNA than what we previously thought. Sorry, getting a drink. So as you can see, DNA is anti-parallel in that you have a 5' to 3' directionality in one strand, and then a 5' to 3' in the other strand. So, and you also get a twisting that you can see here, forming the helix, which also forms minor and major grooves. These minor and major grooves are important because you have different binding opportunities between the minor and the major groove, and you have different drugs. Oops, cancel. No! Okay, hold on. Coming back. I must have hit stop for some reason. Alrighty. Sorry about that, guys. Okay. So again, because of the twist of the double helix, you end up, like I said, with a minor groove and a major groove. And now based on the kind of protein or drug interactions, you may have molecules that prefer to interact with the minor groove or molecules that prefer to interact with the major groove, and they may have different effects on the overall structure and the binding that's going on. So based on Chargaff's rules, as well as Rosalind Franklin's X-ray data, Watson and Crick were the first to propose this double helix structure in 1953. It took a few years before it was widely accepted and popularized in science, and they were also the first to propose that DNA is actually the material that carried genes and hereditary storage. Now, you can have different helices. So the one that we first discussed and the one that was first predicted was the B helix. And it's the most common helix. It's the one we see most often. But you can also have an A helix and a Z helix. Now, A helixes occur under more dehydrated conditions. And you can see there's differences between each of the helices. For instance, in rise per base pair, A has 2.3 angstroms versus 3.3 versus 3.8. And as you can see here, the A is more shorter and squat compared to the B, which is a little more elongated because it has that bigger rise. And Z, which even has the biggest rise, is a lot more elongated. But then helix diameter, we can see that A is a little fatter, B sort of in the middle, our Goldilocks, and then Z is our skinniest. I'll let you go through this table, but the important thing is to understand the differences. Again, A and B are both right-handed helices, whereas Z is a left-handed helix. Z occurs when you get repeats of sequences of GC, GC, GC. A and B can have any various pattern of nucleotides, but we see the Z occur a lot when you get repeats of GC, GC, GC. Okay? Also, the glycosyl bond formation in both A and B, you have anti-puckering versus anti at C and syn at G in those sugar glycosyl bond conformations. So DNA isn't, when it's packed into the cell, you got to remember there's billions of bases in the chromosome of, say, humans. So, we have 23 different chromosomes, right? So that's 46 total because we're diploid. And the way DNA is packed is, if it was just a helix, it would stretch, I think it's like 4 meters long. And obviously we can't just have 4 meters of DNA just in a helix in our cells that are, very, very small. We're talking, nanometer small. So the way DNA packs itself in our cells, say for eukaryotes, is through chromosomes. Now we can get these compact, very tight packing of helices through supercoiling, as we can see here, around histones, nucleosomes, and then as you can see we can supercoil that in order to make chromatin, and then eventually the whole thing is our chromosomes. Hopefully for this, a lot of y'all, this is review, but I just wanted you to understand, again, that supercoiling, as we can see here, is really important in packing all this genetic material, all this DNA, into the nucleosome in our cell, or the nucleus in our cells. It's actually pretty remarkable how the cells can do this. Now you may ask yourself, well, if we're all supercoiled and bunched up into all these small little wrappings, how do we get from this to this in order to replicate the DNA? And that is a whole lecture in and of itself, is that unpacking, uncoiling, and then replication that we're not going to get into, but it's something maybe in a more advanced class we can talk about. Now, RNA. So RNA is primarily single-stranded. It contains ribose instead of deoxyribose. It has uracil instead of thymine. And it can often form much more complex structures. With DNA, we usually just have the duplex helix DNA. However, RNA being primarily single-strand, because it's single-stranded, it can form things like stem loop structures. You can get a number of different stem loop structures that then can form, this is tRNA, so we can see, how more complex tRNA is. This isn't just a simple single strand. And then we can even get very complex if we look at ribosomal RNA. Okay, so the ribosome is made up mostly of RNA with a little bit of protein, but all of this you can see is the ribosomal RNA, and you can see there's lots of stem loops going on, there's lots of bulges, there's lots of, very long-range molecular interactions occurring in order to form this very complex structure. Studies have shown that RNA molecules can actually catalyze reaction, and we actually believe that the very first life forms on this earth were utilizing RNA instead of protein, because it's sort of like the chicken or the egg, right? What came first? If we have to use RNA in order to synthesize protein, then it makes sense that a lot of the beginning creatures, or the prehistoric, that's what I'm looking for, the prehistoric unicellular organisms probably utilized RNA much like we utilize proteins now in order to catalyze reactions in the cell. And then eventually through evolution, proteins came into the world. So DNA replication. So this is just a summary just to kind of remind you of what replication is. We're not going to go into very much detail about it. Again, that's more for a more advanced course than just this, introductory biochemical course. But do know, DNA replication occurs when cells divide, and then each DNA strand acts as a template for the assembly of its complementary strand, such that in the end, you have a new DNA molecule that consists of one parental and one daughter. So you have one old cell, or old strand of DNA that was the template, and you have a new strand of DNA that was formed during the replication. So you'll end up with two strands, or two helices, one with each with an old and a new strand. Transcription and translation. So a gene in DNA is transcribed to make messenger RNA. So here we have transcription to make our mRNA. Then the messenger RNA is translated by the ribosome to synthesize a protein utilizing tRNA. So if here we have our ribosome, tRNA is brought to the ribosome, utilizing the anticodon of the tRNA to the codon of the messenger RNA. We bring the right protein, and we can then start to synthesize our protein using translation. Again, this is something we can go in a lot of detail. We could spend a week talking about this. However, I just kind of wanted to just summarize it for you, just so we can keep it in the back burner of our minds. Now, this transcription and translation is actually called the central dogma of molecular biology. And the book kind of goes into a little more detail about that. A lot of biologists use this term, and immunologists and stuff like that. Chemists don't necessarily use it as often. But the central dogma of molecular biology is that genes and DNA are transcribed in the messenger RNA and then translated into proteins. So that if you affect the gene, you're going to affect the protein downstream. So the human genome was sequenced. I think they finished the sequencing project fairly recently. It was in 2011. They started in 2006, I believe. And it took them that long to sequence the entire genome. And the chapter goes into a lot about how sequencing happened, but for me and for this class, I think the important things are the results that we discovered after sequencing, such as that over half of the human genome consists of repeating sequences of non-coding DNA. So these are pieces of DNA that don't even code for any kind of gene. Of the coding DNA, or the remaining genome, only 1.1 to 1.4% of the genome actually codes for proteins. And that is actually 23,000 possible proteins, which is pretty impressive. However, only a small fraction of those 23,000 proteins are unique to invertebrates. I left off. So really, even, from kingdoms or animal, genuses, were actually pretty similar, because only a small fraction of these proteins are different from species to species. We can see if we compare the numbers from our textbook and this figure here that I took from a different textbook, we can see there's some disagreement. They say that about 4% of the genes are expressed into actual proteins in the cell, and that 70% are non-coding repeats as opposed to around 50%. However, we see another 30% that are other non-coding regions of the DNA that may not be repetitive. And then here we have about 38% of the genome is possibly able to be coded. However, only 4% of that is actually coded in any given specific cell. And it's pretty impressive. So what my research, just to interject here, consists of studying this region here, right? Which is these repeats of non-coding DNA, and how these regions, which are mostly, promoter regions upstream of the coding genes that will activate these genes to be expressed in cells, or deactivate these genes to be expressed in cells. So I'm really interested in these promoter regions and how we can control genetic expression based on these regions in the DNA. Now, if we look at the proteome, so of this 4% of the genome that's actually coded into proteins, or if we take these numbers, the 1.1 to 1.4%, wherever it is, somewhere in the middle, I'm sure it lies, we have our proteome. Now, of that proteome, or that 4% of the genes, 23% are used in just maintaining the genome. So maintenance, expression, and replication. It's a really, large chunk of the proteome dedicated to just DNA stuff and RNA. 21% is in signal transduction. 17.5% is in the general biochemical functions of the cell. And then we have 38.2% are just various other activities. So metabolism, cell maintenance, is about 18%, signaling 21, and 23.5 is maintaining the genome. So if we take this, it's about 60% of the whole proteome is just basically what I would call cell survival. All right, it's the basic materials cells need in order to survive, while 38.2 of the proteome may be what differentiates, say, an epithelial cell from a cardiac cell, or a cell in your lungs from the cell in your pancreas. Because we know all of these cells are different because they express slightly different proteins that give those cells different functions. Like cells in your stomach are going to release certain enzymes that your lung cells are not going to release, right? So it's pretty cool to me. Okay. So mutations are driven by evolution, which is basically natural selection, right? And hopefully this isn't new to all of you. You've talked about Darwinism, at least in high school. And you can have different kinds of mutations. You can have a point mutation, which is, say, maybe an adenine got mutated to a guanine. We know when DNA is transcribed into RNA, so we have DNA to messenger RNA, and when we transcribe messenger RNA, or we translate that to a protein, we use codons, right? So codons are 3 nucleotides, so maybe it's AGAT, okay? And this codon is going to match up to a very specific tRNA with its anticodon, so it would be CTA. Well, it's not T, right? It's going to be yours, so sorry. Because we're talking about RNA. So these are going to match very specifically to a very specific amino acid. Well, if this point mutation, we change, say, this adenine to a guanine, I don't think I can, I can erase. It may take me a minute. Okay. So if this adenine got mutated to a guanine, now all of a sudden, this codon doesn't match up. So now this amino acid is going to be changed to the amino acid that does match the new codon, which will then cause a mutation in the protein, which can then cause this protein to function and fold differently than what it was before the mutation. Now, 99.999% of the time, this may just kill the organism. All right, it may be such a bad mutation that it makes the protein denatured and no longer work, and the organism can't survive. However, sometimes, about 0.1%, it may be beneficial. And now this new protein may give the animal or the organism an advantage to survival. For instance, if we look at this picture, we see we've got green beetles and we got a couple orange beetles. Now, these birds really like green beetles. They only want to eat green beetles. They do not like the orange ones. Well, the orange ones, let's say it was a point mutation in their protein that made them colored orange instead of colored green. Well, now the green beetles keep getting eaten, leaving the orange beetles to mate and produce more orange beetles. And again, as we go through another life cycle or more generations, we see that preferentially, again, green beetles are getting eaten, leaving eventually just orange beetles as our green beetles undergo extinction. Of course, now our birds are probably going to go extinct because they don't have any orange beetles to eat. All right, so we can also get mutations from recombination. Now, recombination is when you have large amounts of DNA or chromosome crossing over. So you can have, say, a chromosome, and then say you can have this whole region of the chromosome crosses over to another chromosome, and you get recombination. And this usually has much more drastic effects than just the point mutation. And this is where you can get disorders such as Down syndrome and much more serious disorders. So usually this may not drive evolution. Transposition is when segments of foreign DNA get added into a host DNA. So this is how viruses work often. They will insert their DNA into the host's chromosome, and then they will utilize the replication and translation components of the host cell in order to spread the virus, and produces the virus's proteins as well as replicated the virus's DNA. Again, these are topics that you may talk about more in genetics, but I just kind of wanted to give you the idea of, yes, most of the time mutations can be bad. They can either go unnoticed, the mutation may not even change the protein, or it's bad for the organism and will kill the organism. But sometimes, that 0.1% of the time, it can be a good thing, and it can drive evolution and natural selection. So really, that's all I wanted to cover in this chapter. Basically, we can go back to our learning objectives and make sure that you can answer all of these for the test, and you should have a good understanding of nucleic acids. Like I said, I'm going to take a minute and I'm going to post the solution to the case studies. I'm going to kind of sketch it out for you so you have an idea about that later this evening after I put my son to bed. So that will come out around 8 o'clock or so. All right. So happy studying, and I will see y'all in class next time. Okay? And let me stop the recording.